currently most people use a number of different assays to capture the different features of gene regulation. Of course, accessible chromatin, which are the regulatory elements, nucleosomes, histone modifications, chromatin conformation, and of course DNA methylation as well. Now, Fiberseq can do, for all of these features that I highlight here in green, all of this you can do from one assay, Fiberseq, which is rather amazing, and relies, of course, on long reads and is PacBio-enabled. Now, you've heard a lot about already how Fiberseq works. It relies on a hyperactive adenine methyltransferase, and all the accessible adenines in the chromatin will be methylated, and then you can read this out with PacBio sequencing, just as you can with the endogenous CpG methylation. In our lab, we prefer to apply the HER5 directly to the isolated protoplast. Of course, you can also do this with nuclei. People often ask me, how hard is it? If you can do ATAC-seq, you can do Fiberseq, okay? It's easy. Before I show you novel biology, let me convince you that we can find known biology, and this is what I'm showing you here. Accessible chromatin regions, this is an aggregate plot, should have a nice peak of accessibility in the center, and this is what I'm showing you here in purple, and CpG should be depleted, which is also what I'm showing you here. Of course, we also expect this really strong signal of accessibility to reside in the transcription start site. Again, this is an aggregate plot here, and you see this strong purple signal, and CpG is depleted. What was very fascinating for me right away when I looked at this plot are these regular ripples downstream of the transcription start site. Let me show you that actually this accessibility signal, both at the transcription start site and at these ripples, corresponds, is strongly associated with expression. The warm colors here, high expression. So the ripples are actually the well-positioned nucleosomes after when transcription occurs, which you can easily see because in the blue colors, where we have low expression or no expression, the ripples disappear. Why is this exciting to a plant genome scientist? In animal genomes, you find this easily with ATAC-seq and DNase-1-seq, but in plant genomes, you don't. Okay, this is due to the much better single molecule resolution that Fiberseq has. And I'm taking the sizes of the methylation sensitive patches apart here to just show you my beautiful ripples. You know, people have obsessions. I apologize. I don't have a lot of time. A picture speaks more than 1,000 words, so I brought you this picture here. This is a screenshot of the Arabidopsis genome with lots and lots and lots of fiber. The methyladenine signal here is in plaque. You see in blue the genome models. And I want to point your attention right towards this promoter region, where you can see that literally every fiber is occupied by probably the core transcriptional machinery. The situation is much more nuanced, shall we say, complex. Favorite word in genome sciences. On the right side, where you really see, for the first time, you get an idea that proteins are stochastically coming off and on. So this protein occupancy for the first time really visualized. And on the right side, again, I show all the features of the epigenome of gene regulation that we can extract via computational analysis from Fiberseq data, including the 3D genome, which I will not show today. Okay, the reason why we wanted to use long reads, the reason why we adapted Fiberseq to Maize and other plant genomes is really because plant genomes and crop genomes in particular are highly repetitive. The Maize genome is 85% transposons, and so I had this fantastic idea that lots and lots of regulatory regions are hiding in these regions that we cannot map. And to do this properly, of course, we had to first very carefully ascertain what regions or how much of the Maize genome is actually mappable with short reads. And this is what I'm showing you here. This is a simulation analysis to avoid any experimental bias. And about only 40% of the Maize genome are accessible with short reads. In our work, of course, we had a paired or several paired ATAC-seq samples, and not surprisingly, ATAC-seq is a short read method, and so the majority of the regulatory elements that we find with ATAC-seq is in this highly mappable region, not surprising at all. Now, the surprise really came from our Fiberseq data. I gave the punchline already away, right? It doubles the regulatory landscape of Maize, but none of that stuff really is in these unmappable regions, right? So still the majority of Fiberseq ACRs or Fiberseq detectable regulatory elements reside in highly mappable regions. We capture quite a bit in these low mappability regions, about 10,000, but really it does not double the regulatory landscape. So what's going on? What is ATAC-seq, the gold standard method? What is it missing? And this is what I'm trying to show here. The element size is indicated by the colors of my graph. On the x-axis, we see the fire accessibility score. On the y-axis, the ATAC accessibility score. And what I hope you can appreciate is that for elements that are rather short, below 300, below 200, actually is the threshold we decided upon, these elements are not detected by ATAC-seq whatsoever. And this is my summary. We have about 107,000 regulatory elements in the B73 genome. Half of these are only detected by Fiberseq. The majority of these are these small guys that are below 200. About 10% of these Fiberseq-specific ACRs come from low mappability, as I showed you. And the ones that are shared, the other half, and literally all of them are shared, we detect pretty much every ATAC ACR with Fiberseq, are the more traditional, longer ones. And this is just a shift control where the chromatin, of course, is closed. Now, many of you may ask, okay, are these real? How come we've never seen them? And what I'm showing you here to just convince you briefly is that they show the same characteristics of regulatory elements which we have known about since the 1980s. We see this peak of accessibility and the depletion of CPG methylation. We have other lines of evidence which I'm happy to share when we meet after the session. So in short, ATAC-seq misses short elements and those that are less frequently occupied, that are more stochastically found by transcription factors. I've already referred to the Maize genome as loaded with transposons. Basically, it is all transposons with some genes interspersed in it. And so we were particularly interested to see, could we actually resolve individual LTR retrotransposons, which are the most frequent class? And the short answer is yes. There was one small subset, about 100 in the B73 genome, that showed this stereotypically presence of putative enhancer and promoter ACRs in both of the LTRs. I'm going to show you an actual real-life example, a screenshot here. I want you to appreciate that there's really no mappability here, there's no ATAC-seq signal here, but we do see these perfectly outlined accessible elements here in the fibers below. And as expected for a bona fide regulatory element, these are hypomethylated, just like they should be. Now, these accessible chromatin regions in LTR retrotransposons come in different flavors. And this one here that I'm showing you is much more common. There's about 1,000 elements like this that show now a single ACR in each of their LTR arms. And as you can see here, they're not quite as pretty anymore. And actually a fair number of them are hypermethylated. This is a truly plant-specific novel epigenetic mark that we only discovered with PacBio-enabled Fiber-seq. It does not exist in animal genomes. And the hypermethylation and the adenine accessibility methylation exist on the same fiber. How that mechanistically works, I have no idea. Now you may wonder, okay, what disappears? The enhancer or promoter, and it is almost always the promoter, leaving the putative enhancer free to interact with whatever other regulatory region they may be able to find in the Maize genome. Some of you may doubt my characterization of LTR retrotransposons, enhancers, and promoters, and our reviewers did too. And so we answered first with this graph that I'm showing you here, that the putative promoters really perform very well with CNN models that we developed from our MPRA data. But I think much more convincing is what I'm showing you here. This is another aggregate plot. We're looking at the putative promoter and putative enhancer. And what I hope you can appreciate is that the putative polymerase footprints are present indeed very beautifully in what we call the putative promoters. We do see transcription factor footprints in the light blue in both the enhancer and the promoter. What also caught my interest right away are these perfectly phased nucleosomes between the enhancer and the promoter. I also want to point out, so these are, remember, these are LTR retrotransposon, probably being in the process of being silenced. These very dense nucleosomes, this high density of nucleosomes, suggests that heterochromatinization might be in progress. So really fascinating. I showed you in the very beginning that well-phased nucleosomes are associated with highly expressed genes. So what we're seeing here is at least consistent with enhancer transcription, which is still a controversial concept in plants. Now, I entitled my talk as Beyond Peaks. And this is really, I think, where Fiberseq and PacBio really shines, that we have these individual molecules and these very complex features underlying what we just call as peaks. So in ATAC-seq, DNase-1-seq, MNase-peak, you just call peaks. What I'm showing you here are these very complex patterns that we find incredibly fascinating. Very sharp, diffuse, complex. And so Morgan Hamm, the student who got really interested in these, just started to derive different features that we can detect with Fiberseq. Of course, the methylation kinds, nucleosome footprints, their positioning, their phasing, polymerase footprints, 19 features in total. Not all of them I want to talk about here. And he built an ACR-centric model. Now, this is a very complex model. Just think you have 107,000 ACRs by 19 features, so you need dimensionality reduction to learn anything. And so I'm going to show you some UMAPs. I know they're not particularly informative, but please bear with me. So this is my first UMAP. And what you should keep in mind that because, of course, gene regulation has both forward and reverse, these UMAPs are symmetric. Yeah? So in blue outline are the LTR retrotransposon ACRs, which in this larger body here, in this large cluster, are all hypomethylated, okay? Like bona fide ACRs should be. And in yellow, or in gold, shall we say gold is nicer maybe, we do see the ones that I showed you as the paired ones, these really very strong, beautiful outlined ACRs. But really, when Morgan came in with this picture, what he was really excited about, that the clusters very much put those ACRs that were hypermethylated on their own. And what you can actually not see, and I apologize for that these clusters contain many, many dots that are not colored in blue, and these are unannotated LTR retrotransposons and LTR retrotransposon fragments. So we can use this now actually for genome annotation. And just to show you that we actually do capture, just with this very simple modeling approach, this curated, manually curated approach, that we can capture these different features of at least promoter ACRs. I'm showing you forward promoter ACRs here again in gold. And as you follow the arrows around, you can see that very specifically these patterns reside in different parts of the UMAP. And of course, we can bring this back to the complex region that I showed you in the beginning. We see our very tightly outlined ACRs, as I indicate them here with arrows. The small one resides in a different part of the UMAP, and of course, we can also find our complex ones. Lastly, I want to talk about hybrids a little bit. Again, I apologize, I'm not a breeder, but hybrids are fascinating. They're twice as big, they make twice as many seeds. Even when you grow them in the dark, they're really huge. And so hybrids have fed the world. And because they're so remarkable, they've been the subject of intense studies for about, at least since 1935. Barbara McClintock had opinions, like she had opinions for many things. And her hypothesis was genomic shock happens and the transposons become active. Okay, let's see what we can see, yes? So I'm going to start with one finding that blew my mind as a genome scientist. And that is that the divergence of regulatory elements of regulatory DNA between two Maize lines, B73 and MO17, equals or actually supersedes that of mouse and human. 43% of the regulatory sites that we can call with Fiberseq in B73 are not present. The sequence, the underlying sequence, is not present in Mo17 and vice versa. Okay? Mouse and human are diverged 100 million years, and their divergence is actually 40%. Okay, so this is, to me, as a genome scientist, not a Maize geneticist, this blows my mind. Among the regulatory regions for which homologous sequence exists in B73 and Mo17, there's over 1,000 ACRs that show clearly hybrid, significant hybrid-specific changes in accessibility. So lots for us here to investigate what that does to gene expression. I should also tell you that we have, of course, kinetics long-read data for all of this. Our transposons are actually expressed, and we have small RNA data as well. What happens with transposons? Turns out that Barbara McClintock was right. So what I'm showing you here are the expected number of silent retrotransposons in the F1 versus observed. You can see that these are going down, and these are the expected and observed numbers, again, for the LTR retrotransposon ACRs that are indeed active. And what was surprising to us, and what I sort of shaded here in blue, is that it appears to be that transposons that are silent, that are fully silent, acquire one active regulatory site newly in the F1 hybrid. And I want to show you just one screenshot where the mom is on top, B73, the hybrid on the bottom. And this is a similarly, or much more frequent, actually, event here, but that is not captured in this table, where we have the two ACRs are already being called, but they become even more accessible in the hybrid. And with that, I'm done. We can double the regulatory landscape of Maize. I'm sure that would happen in your crop as well. We can look for the first time really at the regulatory activity of individual LTR retrotransposons. I showed you that there's a huge promise here to really look beyond peaks, what features actually lead to regulatory activity. We do see very specific features, for example, associated with the 3' UTR. And I showed you that, of course, the haplotype resolution that Fiberseq gives you will, I think, prove really valuable for understanding Maize hybrids better. And with that, that's my group before the funding cut. And these are the people and funding sources that I would like to acknowledge. And I look forward to your question after the end of the session.